Hello everyone, I'm Rafael Blanco Dominguez, and I'm honored to present our latest work about how CD69 expression on regulatory T cells protect from immune damage after myocardial infarction, a study led by the laboratory of Professor Pilar Martín from the CNIC in Madrid, Spain. The relevance of this work is based on the fact that the ischemic heart disease is the #1 leading cause of death worldwide. It is known that inflammation plays an important role in MI, and increasing evidences highlight a crucial role for T cells in the post-MI inflammation and progression towards chronic scenario. So we aim to dissect T cell-derived molecular mechanisms and molecules that confer beneficial properties that could have a therapeutic potential. In a close collaboration with La Princesa Hospital, we received fresh blood samples from MI patients within the first 24 hours from hospital admission. The unsupervised hierarchical clustering of single T cells revealed that a subset of Treg cells that express CD69 on their membrane were expanded in peripheral blood after MI when compared with healthy donors, although in a bimodal distribution, with a group of patients that were unable to upregulate this CD69 receptor. So we aim to study the role of these CD69 positive Treg cells after MI and what are the consequences of a differential expression of CD69 in the progression after myocardial infarction. In order to do that, we moved to a mouse model of myocardial infarction induced by the permanent ligation of the left anterior descending coronary artery. And interestingly, we observed that CD69 deficiency showed a significant decrease in survival after MI. We found that 2 days after MI, as you can see here on the right, CD69 deficient mice presented increased inflammation, increased infarct size, and heart dysfunction measured by echocardiography. Therefore, our data indicate that CD69 expression is important to prevent excessive damage after myocardial infarction. The analysis of the cellular infiltrate by FACS revealed that CD69 positive Treg cells are specifically recruited to the infarcted myocardium. And further analysis revealed that the absence of CD69 allows the accumulation of gamma delta T cells in the heart that secrete interleukin 17, which were described to be deleterious for MI because they boost inflammation and also fibrosis. Then, in co-culture experiments, we demonstrated that CD69 positive Treg cells are key to suppress deleterious gamma delta T cells as they induce apoptosis and inhibit IL-17 production on gamma delta T cells more efficiently than CD69 deficient Treg cells. Mechanistically, we observed that this is a mechanism dependent on CD39, which is an ectonucleotidase that converts extracellular ATP released upon tissue injury to adenosine, which induce apoptosis and inhibit target cells. Our data showed that CD69 drives CD39 expression on Treg cells. So CD69 positive Treg cells exhibit higher expression and also activity of CD39 than CD69 deficient Treg cells. And we proved that when we chemically inhibited CD39, the suppressive properties of CD69 positive Treg cells toward gamma delta T cells were impaired. Ultimately, in vivo, we observed that the adoptive transfer of CD69 sufficient Treg cells 4 hours after MI improved survival in CD69 KO mice, which was accompanied by a reduced recruitment of IL-17 producing gamma delta T cells and a reduced inflammation in the infarcted myocardium. And importantly, this therapeutic effect of CD69 positive Treg cells was impaired in the presence of CD39 inhibitors, altogether supporting that CD69 positive Treg cells in a CD39 dependent manner improve the progression after MI in mice. In order to translate this finding to a clinical scenario, we compared the follow-up of patients with differential expression of CD69 during acute MI, and in agreement with preclinical data, patients with low levels of CD69 positive Treg cells during acute myocardial infarction exhibit higher risk of readmission due to heart failure developed in 2.5 years. These data were validated in 2 independent cohorts of MI patients, one from La Princesa Hospital in Madrid and one from the Santa Creu y Sant Pau Hospital in Barcelona. So in summary, Our data identified that 669 positive Treg cells are key to control cardiac inflammation and damage, boosted by IL-17 producing gamma delta T cells, and high levels of 669 prevents from heart failure development in mice and humans, highlighting this axis as a new prognostic and therapeutic candidate for MI. Thank you very much.